Hi, this video is a recording from the June 16th informational and kickoff meeting of a comprehensive Nooksack basin study of the distribution of bull trout, Dolly Varden, and other trout and salmon species. We're looking for citizen volunteers to collect data at about 300 locations throughout the Nooksack during the summer of 2020. This presentation provides an overview of the science behind the project and why it's particularly interesting to better understand the distribution of bull trout, Dolly Varden, and other species in the Nooksack. I'm Steve Cope with North Sound Trout Unlimited and Fourth Corner Fly Fishers. I'm the local coordinator for the project and organizing volunteers such as yourselves. Our presenter today is Mike Young. Mike's been a fisheries ecologist with the Research and Development Branch of the U.S. Forest Service for over 30 years, and is a co-founder of the National Genomics Center for Wildlife and Fish Conservation in Missoula, Montana. His focus has been the conservation biology of freshwater species in Western North America, with a recent emphasis on their phylogenetics and phylogeography, as well as species distribution modeling and the development of environmental DNA sampling. Mike?
Thank you, Steve. My name, again, is Mike Young. I'm a research fisheries biologist with the Rocky Mountain Research Station, which is part of the Research and Development Branch of the U.S. Forest Service. I'm also one of the co-founders of the National Genomics Center for Wildlife and Fish Conservation. Tonight, what I'd like to talk to you about is using an evolving technology to address one of the most fundamental questions we have about any of the organisms we care about, and that is understanding its distribution. So it's surprising that for as much money and time and effort as we've invested for a lot of the marquee species we care about, even species that are federally listed under the Endangered Species Act, we still have substantial uncertainty about which watersheds these species are found in and where they're found within those watersheds. So tonight I want to talk about that in light of an evolving technology that can help us address that problem, and that is environmental DNA sampling. I want to begin tonight by introducing you to the team that works on environmental DNA sampling within Forest Service, within the Forest Service Research and Development Group. Then I want to describe what environmental DNA is, how we sample for it, and the strengths of that kind of sampling for understanding the distributions of fish and other aquatic species. And then I want to propose your participation in a particular project involving environmental DNA sampling in northwestern Washington. And that is to understand the distribution of 3 species of chars: federally listed bull trout, Dolly Varden, which are restricted to northwestern Washington, and introduced and sometimes invasive brook trout. So first of all, I want to begin by introducing the folks from the National Genomics Center. On the upper left-hand corner, you see Kevin McKelvey, and in the upper right-hand corner is Mike Schwartz. Those are the 2 other co-founders of the National Genomics Center, and Mike Schwartz is the current director of the center. Underneath Mike is Kelly Kareem. She was our first environmental DNA program coordinator and is currently our tribal liaison and is leading the range-wide eDNA-based Pacific Lamprey project. Beneath Kelly is Tommy Franklin, and all the way over in the left corner is Jennifer Hernandez. Those are our current co-coordinators of the environmental DNA program. And then above Jennifer is Taylor Wilcox, and he is single-handedly is responsible for most of the technological advances that we have made at the National Genomics Center. We also have a whole series of colleagues at the Boise Spatial Streams Group who are members of the Boise Spatial Streams Group, led by Dan Isaac. And these are folks who are specialists in spatial data analysis, in database design, and in website design, including Dave Nagel, Donna Horan, Sherry Walrab, Sharon Parks, and Gwen Chandler. So one of the reasons we got interested in doing environmental DNA work is that we're trying to use conventional sorts of sampling to try to answer questions that conventional sampling really wasn't good at. And that is for sampling some really rare species like loach minnow and spike bass in the Southwest, which are federally listed under the Endangered Species Act. It's actually regarded as harmful to use traditional forms of sampling to try to capture them. And generally, the traditional forms of sampling we like to use are electrofishing, and in many cases, those are prohibited. In any case, even when electrofishing is really effective, it's always time-consuming and sometimes relatively expensive. That's because it typically involves 2 or 3 people. At best, they can generate abundance estimates or even detect species over about 200 or 300 meters in a day very reliably. Therefore, they really can't cover the tens of kilometers that typically constitute your average good-sized stream virtually in any part of the western United States. And then finally, there are conditions when the animals we care about are really hard to find. That is, when they're really rare, and that can either be because that's the native state of how many of our fish populations are. Some of our natives are really rare a lot of the time. But also we may be trying to detect invasive species just as they're arriving. So we're trying to find the invasion fronts or to remove survivors of chemical treatments. And for this kind of work, traditional electrofishing is just not very effective. So what we were hoping for was that there was an alternative. And that alternative turns out to be environmental DNA sampling. Now, environmental DNA is just DNA that's been released by a fish, by a bird, by an insect, or by you into the environment. And it's that environment that we sample in the hopes of capturing that DNA to infer the presence of that fish, that bird, that insect, or you in the environment. Typically, when we do this kind of work, what we're looking for is mitochondrial DNA, although not always. Sometimes we will look for nuclear DNA. We typically sample in streams, and a lot of environmental DNA sampling has been done there, but quite a bit of it lately has also been in wetlands and in ponds, both to look for fish and to look for amphibians. And as I say, it usually is for fish that we are trying to do these kinds of surveys, but environmental DNA sampling can be done in a whole host of environments for a whole range of taxa, and I'm going to show some of you those in a minute. Now, the reason that we've become such advocates for environmental DNA sampling, at least the way that we practice it at the National Genomics Center, form three legs of a stool, if you will. First of all, eDNA sampling, the way we do it, is really fast. It typically takes 8.5 minutes to collect an environmental DNA sample. It takes a couple minutes to set up the equipment and a couple minutes to put it back away. So all told, about 15 minutes at every given site. In the upper right-hand corner there, you can see all the equipment that's involved. It's not very much. It all fits into a backpack. And what that means is that every person listening to this presentation is an entire environmental DNA sampling crew. Moreover, what we're saving here is a filter, not water. And what you see in the center of the slide is that filter folded inside a plastic bag. That plastic bag has desiccant. And also, that plastic bag is the data sheet because you don't need to record very much information with any one of these samples. And then once we have that sample in that desiccant, it's actually shelf stable. All we do is put it in a paper envelope to block light and then throw the whole thing in a cooler where it can be stored for weeks. So we don't have to immediately get it to a freezer. And it's because of this speed and this portability that rapid broad-scale surveys of aquatic organisms are now possible. And so it's great that eDNA sampling is really fast, but what's even more exciting is that it's extraordinarily good. And it turns out for detecting most species in most kinds of aquatic environments, environmental DNA sampling is both more specific and more sensitive than traditional sampling for a whole host of taxa and circumstances. Consequently, the occupancy estimates that we derive from environmental DNA sampling are highly robust. That literally means you can take them to court because they're likely to be extraordinarily reliable and better than traditional sorts of estimates. And not only can you use it to determine the presence or absence of a species, but it's increasingly being used to provide some information about the abundance of particular species at particular sites. And so the third leg of this stool that makes it truly revolutionary is that it's really easy to learn. It takes me about half an hour to teach somebody to become an environmental DNA sampler, and after you collect 2 or 3 or 4 samples, you literally are an expert at that point. And that makes the technique crowdsourceable. And when it becomes crowdsourceable, that means it's not just what I'm interested in that matters or what someone else is interested in, but there's a whole host of stakeholders who now care about individual species. And that's what makes the truly revolutionary part possible, and that is we can do near real-time, whole range-wide assessments of the status and trends of individual species. It's something I've never been able to do in my scientific career. And so there are 3 legs to also the stool of what you do with an environmental DNA study or that support or underlay that environmental DNA study. And first of all is the protocol you use. and the way you collect the samples. The second, though, happens in the laboratory, and that's based on the quality of the individual quantitative PCR assays, qPCR assays for short. That's what the lab folks actually use to determine which species is present and only which species. That is, they should be absolutely specific to the species you're focused on and extraordinarily sensitive to the presence of the DNA of that species. When I say extraordinarily sensitive, what I mean is typically in our reactions, if a single copy of the target species is present, that DNA will amplify and it will generate a positive detection of that species. And as you can see, we can do it for a whole host of taxa. It's not just the charismatic trout or chars or salmon, but it certainly does include all of those. We've actually got a generic marker that can tell you that any salmonid is present, so it could be anything there. It doesn't have to be salmonids. It can be the non-bony fish like lamprey. It can be other game fish that we care about or invaders that we worry about like smallmouth bass or northern pike or sauger or walleye. It could be a whole host of non-game fish, and I've got a particular fondness for sculpin. For those of you who know, I've got a huge project identifying the molecular diversity within sculpin in Western North America. We do this kind of work for amphibians, we do it for mussels, we do it for invertebrates. We can do it for aquatic mammals like otters or a whole host of aquatic fish. And you can see the list of species there on the right side as well. If you can imagine it encountering an aquatic environment, we can design an assay and a study to sample for that species. And there's an aquatic environment that most people overlook, and that is snow. And so we've developed an entire sub-program that samples snow to detect the presence of forest carnivores like lynx and fisher and wolverine. And then the third leg of any good environmental DNA project is the engagement of the stakeholders. And to that end, what we have developed is a very simple but robust protocol for eDNA sampling in streams that's posted online. Anybody can see it. And this protocol has been beta tested at this point by literally hundreds, if not thousands of biologists. So the errors, the wrinkles, the warts in this thing have been worked through and more or less ironed out. We also provide support in the form of a website that provides a whole host of information about environmental DNA sampling. But more to the point, we serve as a lending library. And that is, if you're working with the National Genomics Center, you can get all of the equipment and supplies you need to conduct an environmental DNA survey project for your particular species of interest. And it's because of our willingness to do that we've engaged a whole host of individuals from state and federal agencies, from municipalities, from industry, from NGOs, tribes, and academicians. And the point being, we literally have and will work with anyone who is interested in conducting an environmental DNA survey project. And so there are a bunch of applications I'm sure you can imagine for using environmental DNA. I've already talked about a couple of these things. That is, maybe finding the invasion front for things like brook trout or smallmouth bass, or ensuring that chemical or physical treatments to remove those species have been successful, so looking for the last survivors of those kinds of things. It's increasingly being used to monitor migration, to know when species are arriving at particular locations. But the application that is truly in the wheelhouse of environmental DNA sampling is the detection of rare native species, for which the bull trout have to be the poster child. So bull trout are federally listed under the Endangered Species Act as threatened, so their presence or their absence has an influence on both land and water management. Although bull trout are extraordinarily widespread, almost everywhere that they're found, they're also rare. So they are difficult to find using traditional methods. Another little wrinkle that makes them useful or very interesting to design surveys for is that the juveniles are really constrained by their environment and by climate. So it's easy to target surveys to find bull trout. And so we did a test in 2014 in Montana to see how well eDNA sampling would stack up against traditional sampling. So what you're seeing here are 4 panels. The top 2 panels involve sampling in one stream in western Montana. The bottom 2 panels involve sampling a different stream. The panels on the left involve data derived from electrofishing. The panels on the right, data derived from environmental DNA sampling. So in the upper left-hand corner, the filled squares indicate locations in a stream where bull trout were detected using traditional electrofishing. The open squares indicate locations where bull trout were not found To the right of that is comparable data for environmental DNA sampling. Filled circles where bull trout were detected, open circles where bull trout were not. Virtually the only difference between those two panels is that environmental DNA sampling found bull trout a little bit farther upstream in that basin, but there's one other huge difference. It took 15 years to gather the electrofishing data. It took a colleague and myself until 2 o'clock in the afternoon on a single day to collect the environmental DNA data. And that's the true power of the technique, the rapidity with which you can collect really robust data. And if you look at those lower two panels, it's a little bit different here. In this case, the black squares on the left are places where bull trout were not found by electrofishing, red squares where they were found. On the right, the eDNA sampling, black circles not detected, red circles they were detected. Again, everywhere that bull trout were found by electrofishing, eDNA sampling also found them. But the thing that was really important here is eDNA sampling discovered a population in a new location that no one knew about, and that's what's indicated by that arrow as well. So this kind of work got us really excited because at this particular time, we had just built a species distribution model called the climate shield model. And we used that to define more or less a climate and environmental envelope for the distribution of habitats that were likely to be occupied by spawning and rearing segments of bull trout populations throughout the lower 48 states, as well as for cutthroat trout. And we were using that model to make predictions about where bull trout should be today, as well as projections about how the distribution of bull trout might change as a consequence of climate change or a list of other actions. And the first thing that we observed is that we found over 3,000 potential bull trout habitats for which we had very little or no data with respect to whether bull trout were actually present in them. And there's no way we're going to conduct traditional sampling at that scale to try to find out what proportion of those habitats were actually occupied and to validate the model. And so from that was born the notion of why not use environmental DNA to sample this thing across its entire range. And so that's what we did over the last 5 years. We have been working on this range-wide eDNA bull trout survey. And we have engaged hundreds of biologists in thousands of locations. As part of the range-wide project now, we have over 6,000 new DNA sites specifically dedicated to determining the presence or absence of bull trout. And that got us thinking even more. If we can generate that kind of data for bull trout, or if we have that kind of data for bull trout, wouldn't it be really important for us to share that with all the stakeholders outside of our immediate group so that they could see where bull trout were in some of these areas and so that they could conduct their own specific assessments of bull trout status or be directed to new areas that they might like to survey to understand the distribution of bull trout? And then we thought, why restrict it just to bull trout? Why don't we do this for everything that we handle to the National Genomics Center? And so from that thinking, the Environmental DNA Atlas was born. And the Environmental DNA Atlas is quite simply a portal. It's an online open access database to the results of all the environmental DNA samples that have been processed at the National Genomics Center. And as you can see there, we've got data at this point, I think, for over 40 different species. There are over 20,000 samples that are represented within the eDNA Atlas, and we are adding new samples annually. In fact, we're going to upload another 1,000 to 2,000 in about 3 weeks. So this is a rapidly growing database designed to make it easier for the stakeholders to get access to this information about the species that they care about. And so that brings me to the stage of asking you whether you would like to get engaged in a project in a basin that is near and dear to the hearts of many of you listening, as literally hundreds of other biologists have across the West. Specifically, what we would like to do is extend the range-wide bull trout project to the Nooksack River Basin. And it's because there's a very unusual pattern there that involves what I'm calling, I'm sorry for the pun here, mixtures, because it's a place where 3 chars co-occur. And it might be one of the few places in North America where 3 chars are known to co-occur, at least in the lower 48. First of all, there are bull trout, and bull trout are relatively widely distributed within the Nooksack River Basin, but of course they are federally listed, and as a concept, that has consequences indeed for how land is managed, how water is managed, as well as how recreation might be managed. So where they are matters, and where they're not also matters. The interesting wrinkle here is that the southern subspecies of Dolly Varden is also present. So, Salvelinus malma lordii. What's odd about Dolly Varden in this part of the world is they're thought to occupy exactly the same kinds of habitat that bull trout do. But that when bull trout are present, the two species partition the watershed such that bull trout often tend to be anadromous or fluvial or adfluvial and grow to very large sizes, whereas Dolly Varden tend to be very small, adopt a resident life history, and quite often, oddly enough, are found above what are thought to be barriers to fish migration. But they're really, really hard to tell apart. And what I'm showing you there is apparently the only characteristic that reliably distinguishes them, and this is, the arrow is pointing to a branchiostegal ray. And bull trout are thought to have more branchiostegal rays and Dolly Varden fewer. The problem is very, very occasionally the two overlap, and they're effectively indistinguishable. In fact, they were proposed by the Fish and Wildlife Service in the Federal Register to be regarded as virtually, or to be managed virtually in the same way that bull trout would be managed. And this was under the similarity of appearance clause, simply because they could not be reliably distinguished from bull trout based on visual observation alone. You can discriminate them genetically. The thing that's kind of an interesting wrinkle here is that the state record for Dolly Varden is from the White Chuck River. It's a fish that's supposed to be 10, almost 11 pounds. Oddly enough, the range map you're seeing here is from Wydowski and Whitney, suggesting that maybe Dolly Varden aren't supposed to be found in that part of the basin. And more to the point, this is a really big fish, and there are bull trout in that basin. There are bull trout that have access to the ocean in that part of the basin. So based on conventional wisdom, Dolly Varden aren't supposed to be exhibiting that kind of life history. And so the thing that I find kind of ironic might be is that the state record Dolly Varden might actually be a bull trout. And then finally, there are introduced brook trout that are present in portions of the watershed. And the problem with these is they are problematic for both of these species. Brook trout can hybridize with both Dolly Varden and with bull trout. And it's not so much that they lead to intergress populations and hybrid swarms as that they represent wasted reproductive effort for both of the native species. And both of those native species tend to be somewhat rarer than introduced brook trout can be. And we do know that at least in inland populations, brook trout are capable of displacing bull trout from portions of watersheds, particularly the headwater portions of watersheds in very small channels, because they are, in fact, brook trout. So what we're proposing is to conduct environmental DNA surveys at specific points throughout the Nooksack River Basin. And if you go to the range-wide eDNA-based bull trout survey website, you can download maps, not only of the Nooksack Basin, but of any of the other basins where these samples can be done or have been done. And you can look at those maps to see what we gauge to be the probability of occupancy of different reaches within the Nooksack River Basin. And so what we're going to be asking folks to help us with is to sample those sites that are on public lands. And you don't have to go find new sites. All of the sites are already pre-assigned. In fact, we've got Excel spreadsheets there that specify the exact coordinates of each one of those sites. We will be asking folks to try to go to all of the sites that we assign and get as close as you can. The fun thing here is that this literally is horseshoes. Being close enough counts. You don't have to be at the exact point. Often being within 50 meters is great. As before, we're going to provide all the equipment that folks need in order to conduct these environmental DNA surveys. And at that point, we're just going to ask you to either go for a walk or to take a drive. And one thing I wanted to mention to you right now, when we were in discussions with Steve about this project and the things that we might do, we actually ended up developing a new technology here at the National Genomics Center that enables us to examine a whole host of species simultaneously for roughly the same cost of looking at 2 or 3 species. And the thing that's important about an environmental DNA sample is that it has the DNA of a whole host of species on it, and the sample can be used repeatedly until it's exhausted to ask questions about the presence or absence of these species. And it's not just restricted to any particular group. Moreover, these samples can be retained virtually indefinitely. I said before that once they were dry, they were shelf stable. Once we get them to a freezer, they should be stable literally for centuries, quite possibly. And so one of the goals that we are trying to pursue is to find additional funding such that we can build a chip, if you will, at our laboratory. And this chip is loaded with environmental DNA assays. And what we would like to do is examine each one of the samples that you could collect for the presence of all the possible salmonids that are found in the Nooksack Basin. So all the species of salmon, steelhead, coastal cutthroats, I think even mountain whitefish, if that's there as well. So with that, I'd actually like to turn this over to Steve, because this is how you can get involved in the project. And Steve is going to be our point person for this particular area. What we hoped we could do at the end of this summer was to have a huge BioBlitz, where anywhere between half a dozen to a dozen of us would come out from the research station. We would camp on the Nooksack. We would invite all of you up to meet with us. We'd gather together, assign different places to go, and then go out and sample over the course of a week. And we pretty much finished the basin because of how fast you can often go. And if we hadn't finished it, it would only take another week or two for local volunteers to mop things up. Well, that kind of meeting doesn't really seem like it's in the cards given present circumstances. A couple of us may still try to get out there and assist and at least meet some of you in a socially distant and friendly way. But for now, what we're going to have to do is rely on you as volunteers to carry this project forward until we can participate in a more comprehensive way. So with that, I'd like to say thanks for your attention this evening. Thanks for considering participating in this project. And Steve, I'm going to relax control of the screen and turn it over to you.
Thanks, Mike. So if that sounded really interesting to you guys and you're interested in volunteering, what we'd like you to do is send an email to nooksack.dna@hotmail.com. This is an email address specifically for this project and for coordinating volunteer activities. We'd like you to send us your name, email address, and phone number. And we'd like to know a few more things in order to minimize the number of back and forth emails to coordinate schedules on this, because we do have a fairly good response already in terms of volunteers and a limited number of test kits that we can have out at any one time. So we'd like to know the number of consecutive days you're interested in sampling, if you're interested in just doing one day, or you'd like to do a 3-day weekend of it, or something else. If you have some preferred days that really work for you well, or some days that really don't work for you, let us know that. If you have a preferred stream reach, some people have a place that they really like to fish and they'd really like to learn more about it. So one of the ways to learn more about it would be to collect some samples and see what the DNA results are for those areas. We'd like to know where you live. Are you in Bellingham or Sumas or Glacier or Snohomish, or and how far are you willing to drive? So that'll help us figure out, if you didn't specify a stream reach of interest, we can assign an appropriate one for you. We're also interested in knowing what kind of sample sites you prefer. There's a lot of different options in this basin. There's a lot of points that actually fall really close to an existing paved road with a short walk. There's quite a few that are close to four-wheel drive roads. And then there's some other points that require some, we'll call hardy bushwhacking. So we're going to try to figure out an appropriate assignment of points for everybody so that you're not stretched beyond what you're really interested in doing. And finally, your organizational affiliation. This is optional. The project is locally supported financially by North Sound Trout Unlimited, Washington Council Trout Unlimited, and Fourth Corner Fly Fishers, but by no means do you need to be part of one of those organizations to participate in the project. That was just necessary in order to collaborate with the Forest Service and make it happen for the Nooksack. We're really interested in trying to get anybody in the community who's interested in citizen science data collection and understanding species distributions within the Nooksack to come out and participate and help collect data and be part of this really interesting project. If you want to keep up to date on what's happening on the project, we'll be posting information at northsoundtu.org/DNA. It'll also have this bulleted list of things that we'd like in your email, as well as the email address and this video and other information as it becomes available, including the training video, which should be available later in July. Thank you for watching tonight, and we look forward to hearing from you and seeing you out on the river. Thanks. Bye.